For thousands of years, it has been the traditional land of the Huron-Wendat and Petun First Nations, the Seneca, and the Mississaugas of the Credit River. Today, Toronto is home to Indigenous peoples from across the SickKids is committed to working toward new relationships that includes First Nations, Inuits, and Métis peoples. And it's grateful for the opportunity to share this land in caring for children and their families. Okay, so the talk today is going to have a little bit of an introduction when I'm going to explain to you what I do. So we're going to go through main topics of my work. So we're going to start talking about neurological and psychiatric symptoms that are associated with autism spectrum disorder. Then we're going to talk about the brain network governing these behaviors. And then we're going to talk about two types of neurostimulation that I've been doing at my lab. So one is deep brain stimulation and the other one is focused ultrasound. Then we're going to talk about three key nodes of the frontal limbic striatal network, the nuclear accumbens, and how we've been working, modulating these areas to improve symptoms. And we're going to finish with some featured directions that my lab is taking for the next couple of years. So to start, we need to talk about what is autism spectrum disorder, and this is a neurodevelopmental disorder that has two main characteristics, so social communication deficits and repetitive behaviors. In the past, they used to be divided into three different types of difficulties, but now with the new revised DSM-5, we have those two domains. But that is not all. In reality, up to 50% of the children with autism spectrum disorder will show neuropsychiatric symptoms. This is an umbrella term for neurological and psychiatric symptoms. So we put together as neuropsychiatric. And here I put some of the most frequent ones, so aggressive behaviors, anxiety, mood disorders, but they also have seizures. And the ones in red are truly the ones that I'm interested mainly in working with. So it's this more psychiatric side of the neuropsychiatric symptoms. Those children that are affected by those neuropsychiatric symptoms, they have a very negative impact in their academic lives. There is a reduction of the quality of life. And there's severe side effects of medications. We cannot give anything to children. There are lots of medications that are exclusive to adults and they cannot have it. And several medications that they are allowed to have don't have class 1 evidence of efficacy. So we don't even know if they're actually working. And because of all of this, there is an urgent need to better understand the neural networks that are underlying these symptoms and explore new treatment options for these children that are suffering from the symptoms. So to study that, I use mouse models of autism. I do a lot of human work, and that is in collaboration with other centers. Pulse generator will send the electrical signals that are delivered in the brain, and that will change cellular activity and behavior. This is commonly used in Parkinson's disease and other movement disorders, at least in North America, and it has been done in more than 1,000 patients. In psychiatric disorders, this is a potential treatment for patients that do not respond to other treatments. So there is an idea that when the patient is refractory, so it does not respond to behavioral or medication, then they could be eligible to neuromodulation. So we're talking about the most severe cases, those where we tried everything and they didn't improve. But there are very promising results coming, mainly from the adult literature. So to study that, I use a mouse model of DBS, which is a miniature of the human model. So we do have electrodes implanted in the brain. Those are connected through cables through a pulse generator, and we can modulate this electrical activity. And here's a photo of a mouse connected, being stimulated, so they're awake, they're freely moving, they can eat, they can sleep, they can drink, they can do whatever they want. So they connect for a few hours, and then we disconnect them, and we can treat them this way. But then when I was reviewing the literature, and this Christina, one amazing PhD student of mine, we realized that the field is so large of DBS, but we don't really know where we stand in percentage of studies. So she did a systematic review to look at both rat and mice studies to understand where is DBS being studied. Because we can apply DBS to so many things, and there's so much we need to learn. But it's also is DBS being used in the fields that we are questioning? Is this something that has the potential to bring new knowledge? And that we saw that when we look at neurodevelopmental disorders such as autism, we have like 2% of rat studies and about 7.8% of mouse studies. This makes sense because we have more mouse models of autism than rat models of autism, so it makes sense that it's more in mice. But that is very little. Put viruses through the blood-brain barrier without having to inject the virus through the brain. And then we can start with pharmacogenetics and etc. That gives the opportunity of putting drugs into the brain that will not go in. And even if you don't inject anything, some groups already shown that the neuroinflammation that you cause by opening the blood-brain barrier is enough to change neuronal activity temporarily. So there are lots of things that we need to learn about focused ultrasound, and there is also a lot of opportunities coming with that. And then at the Mouse Imaging Center, those two are my colleagues at the Mouse Imaging Center. We're a group of 3 PIs, so Dr. John Sled and Brian Nyman and me. We are together at the Mouse Imaging Center, and this is our 11.7 Tesla MRI. And I have a focused ultrasound system inside the MRI. So I can do ultra-high resolution MRI at the same time that I'm treating, and then I can see the blood-brain barrier opening. For example, when you look at those white dots, those are all areas that we open the blood-brain barrier in a mouse. So we have the technology now to start exploring this. Our hospital is acquiring focused ultrasound systems, so this is a very, very exciting new venue of stimulation and treatment that we are implementing at SickKids. Okay, so that was a long introduction. Let's bind this all together. So I talked to you about the frontal limbic striatal network. I talked to you about DBS and FUS, and I talked to you about neuropsychiatric symptoms. So now let's start combining those things. So if we get the frontal limbic striatal network and the neuropsychiatric symptoms, then it comes my first research question. So what are the precise contributions of each brain region of the frontal limbic striatal network? As I mentioned, we target one, but is this affecting all of the others? Is this the best area? Is this the one that is the most involved in that specific behavior? Then if you combine neuromodulation with neuropsychiatric symptoms, then comes the question, what are the mechanisms underlying symptom improvement following treatment? Not everyone improves the same. Some improve more, some improve less. The question is why? How can we make this better? And then we combine the neural network and neurostimulation, then we're talking about target stimulation. How can these therapies be further developed to maximum efficacy with minimum side effects? Are we choosing the best brain target for everyone? Or different type of patients require different type of treatments or different brain areas? So this is what my lab has been working on, what I have been working for my life, and those are the lines of research that I'm doing. So if I integrate neuroimaging, we can then unlock treatment strategies for pediatric brain disorders. And let's start with the hypothalamus. So the hypothalamus is a key area in emotional control and homeostasis, and it has been historically targeted for treatment behavior, mainly through ablative lesions, so destroying the tissue. This goes back to the 1940s, 1950s. There was a boom of psychosurgery during the 50s and 60s, and there was a lot of studies targeting the hypothalamus to reduce aggressive behavior. So nowadays, we're trying to get away from lesioning the hypothalamus because it's quite delicate and there's a lot of mortality associated with it, and going more into looking at stimulation of the hypothalamus, which is a reversive technique, which is much more safe. So here at the top, we have, it's a computed tomography, but it's like an X-ray, and we can see the electrodes going into the brain of the patient and the cables that will connect this to the pulse generator. And here is a reconstruction of those electrodes. So when we want to study, we take the MRIs of the patients and we reconstruct those electrodes and we calculate this yellow area. It's called the volume of activated tissue. So how much of the brain is having a biological effect from that stimulation. And through that, we can then study how this is impacting the brain. So first thing is, does this work? And the answer is yes, it does. It reduces aggressive behavior. So if we look at the gray bar here, this is the patient before. His total aggression scale was 10, which is very high on that scale. And then at the last follow-up is almost at 0. So yes, it does. It fluctuates. And that is very interesting. And that's one of the questions. Why? Is the simulation parameters the best? How do we adjust this to improve? And we also improve quality of life. And then when we went to the brain to look how this volume of activated tissue is affecting the functional network, so the functional connectivity of the brain, we saw that effective stimulation, so the one that leads to symptom improvement, is associated with an activation of several areas of the frontal limbic striatal network and several areas responsible for the production of monoamines. Remember that at the beginning I said that it was a balance between dopamine and serotonin. So yes, we can see that we are engaging those areas. So we have, for example, the dorsal half nucleus that will produce serotonin. We have, for example, the substantia nigra and the ventral tegmental area that do produce dopamine. So we're seeing that the benefit that comes from DBS, it's also leading to a brain change that engages those areas. But this was one patient. So my question was, is it the case, or is that something that we're going to see constantly with all the patients that come? So went abroad, start getting less aggressive, because now the DBS was starting to do its effect. And those who were not attacking because they had the DBS working, now start to get aggressive. So with that, I saw that hypothalamic DBS reduces aggressive behavior by engaging the frontal limbic striatal network, both functionally and structurally. That this effect is at least partially mediated by serotonin. So now we're going to move into the amygdala, which is the second key player of that story. So the amygdala, like the hypercontrol, and it also has been targeted for reducing aggressive behavior in ablation type of treatments. And that's still done. It is not something that is frequent, but it's done. And the reason why the amygdala gets ablated and not gets DBS, it's because if you stimulate the temporal lobes chronically, you induce seizure. So DBS of the amygdala is not a good idea. So the amygdala is not a good candidate for DBS. So if we want to target the amygdala to reduce aggressive behavior, we need to think of other treatments that are not DBS, not electrical stimulation. So during my PhD, I followed a very rare group of 4 patients who received amygdala lesions to reduce aggressive behaviors. So those are patients with autism spectrum disorder who are extremely severe, so they do not respond to any treatment anymore, and this was given as a last chance, like a humanitarian action to help themselves and their families. So my questions were, one, does it still work? Because there is a little bit of a difference between the research done in the 50s and now. So the question, the second, how do we engage the network? What is this causing in the brain? So here we have reduces aggressive behavior, and that is leading the brain to a different morphology pattern that is closer to normality. Now we're going to go for the last brain area, which is the nuclear accumbens. That's the most recent one. This is a work, as I mentioned before, it's parallel to clinical trials. So here we have both mouse and human work going on. The majority of this work is done by Christina, my PhD student, who is amazing, and she's been bridging both worlds. And the thing that is very interesting is that even though self-injurious behavior is a type of aggressive behavior, it is very repetitive. So for self-injurious behavior specifically, we believe that the nuclear accumbens is a better target because we are working with self-reinforcement of behavior. So this is a mouse model of self-injurious behavior. So what they do is that they self-groom all the time. So they're always licking and pulling and biting themselves. So they pull off hair out of their bodies, they break their skin, and what we see is those bald areas. Sometimes they bleed and then we need to treat them, but when it's just pulling the hair out, that's what we see. So when we do deep brain stimulation of the nuclear accumbens, we can see here the animal that was DBS off. This is at the beginning of the experiment, and this is at the end. It got even worse. But when we got animals and we turned the DBS on and we did the treatment, the nuclear accumbens stimulation, we saw that they improved. So the behavior stopped, and because the behavior stopped, the fur could grow again, and they improved. And we obviously measured that, and we saw that the physical injury really reduced in the aggressive group, and also the repetitive behaviors. So now we're saying about a core symptom of autism, the repetitive behavior, and a neuropsychiatric symptom associated. So we scanned those brains after the treatment, and comparing the brains of the animals that received DBS on versus animals that were in the DBS off group, we again saw a lot of brain changes, so brain volume changes in that frontal limbic striatal network, areas controlling monoaminergic system, and we saw that the change in behavior and change in brain volume were associated for a couple of key areas like the amygdala, like association cortex, hippocampus, and somatosensory area 1. So we are again involving changes in the network, and the change in the network is associated with improvement. So then the question was, are we checking this also in the patients? Or is this exclusive of mice? Because there's quite a lot of difference between the brains of humans and mice, and we need to take that in consideration. So we did the same technique that we did in the mice, that we did in my previous study, the DBM analysis we did in the brains of the patients before and after DBS. And then again, we saw several areas that we detected in the mice, we also detected in the humans of change. So again, we're talking about the frontal limbic striatal network, and it is what it is. It just always keeps activating these areas. So we have morphological change. This is volume change. The question is, does it change function? Yes, it does. So again, we did the reconstruction of the electrodes. This is all the patients together. We calculated the volume of activated tissue for each one of those electrodes for each patient. And then we looked at the functional connectivity, and we saw engagement of those same areas. So again, and then I looked at the brain of the patients who got the surgery before and after, and compared them with those control patients. And I did that by looking at their cortical thickness. And what I saw is that at baseline, this is the difference between the patients who have aggression compared to the control before the surgery. You see several areas. All of those colorful areas are areas that are different. Down here we have the names. But I just want you to visualize, like there are several areas of the brain that is actually different. They have a different cortical thickness, either thicker or thinner, but they are there. But then on the follow-up, which is after the surgery, we see that lots of those differences disappear. So it seems like when we remove the amygdala from that game, we are changing the brain in the direction of normality. So we are not only improving behavior, it seems like we're improving morphology. But as I mentioned, we can't do electrical stimulation of the amygdala. I won't do it. I know there's some people doing, I don't agree. Because I know that in the lab, it's one of the models of inducing epilepsy is kindling electrical stimulation of the temporal lobes. So I'm here thinking, how do we target the amygdala in a way that we don't destroy the amygdala, but we inactivate the amygdala? And to do that, I collaborated with Dr. Lee Banks. She was at Sunnybrook with me, now she's a PI at University College London, and she developed those nanodroplets. So they're like those microbubbles that I mentioned before, but smaller. And she made it in a way that she can put an anesthetic inside these nanodroplets. So when we inject this into the mouse, the anesthetic is in the droplet, so it's traveling in a bubble. When we deliver the focus ultrasound to a specific area of the brain that we want, those bubbles vaporize, and then the drug that was in the bubble gets released. Here, because I'm using an anesthetic, I don't need to open the blood-brain barrier because the anesthetic crosses the blood-brain barrier on its own. So what we are doing here is that we are bringing the anesthetic, a very small dose of anesthetic, to a brain area that we want, and we're releasing only there. And that anesthetic go in and anesthetize the region. And then we did this by targeting the habenulum, acting those brains, doing Western blotting, and etc. So there are lots of techniques that we can do when collecting the tissue. But I am the mouse imaging center, so I want to transform this also in imaging. So we're going from an invasive type of research to also having a non-invasive. So for example, using functional MRI and magnetic resonance spectroscopy to look at those activations, to look at the dopamine concentrations, glutamate, GABA, etc. So that we can have both after treatment and longitudinal. As I mentioned, my focused ultrasound system is inside the 11.7 Tesla. That is the only system that is inside an 11.7 in the whole Canada, and I do not know abroad how it is, but I know that in Canada we are the only one, and I have not seen any publication, which means that every protocol that is out there, we need to adapt, and that's why this is a development stage, because now we need to get what has been done in other field strengths and make it work for ours. And the last and very exciting field that I'm looking into exploring is virus transfection with the focused ultrasound. So going for a completely non-invasive. Opening the blood-brain barrier to have viruses go into the brain. This has already been done in a couple of groups. Researchers, South Korea is very big in focused ultrasound, and they develop a couple of protocols to do this. So instead of injecting the virus through anesthetic surgery, you would use the opening of the blood-brain barriers for the virus transfection. And then we could then add viruses that are those mechanoceptor types, the ones that open with focused ultrasound, or we can do virus that responds to a specific drug and do some sort of non-invasive chemogenetics. So this is definitely something that my lab will pursue. It's not something that I'm going to do it right now, but this is coming years. So to finish, I would like to thank you all for listening and for being here, Eduardo for inviting me, and all of you for an amazing reception, all of my collaborators and funding source. Thank you.
Are there any questions from the audience? Don't be shy. There's one.
Mm-hmm. Hi, impressive work as always. I just was wondering about, there must be a functional rearrangement in terms of how the brain is connected itself. And overall, with amygdala, we know the extended amygdala, and all these changes, it regulates some other process, like memory, like stress deals, how the rats or how the kids deals with the stress. And I just was wondering about the side effects or the possible side effects of this DBS, and if you're going to explore in some time about what could be these side effects.
Thank you, great question. So when we looked at the amygdala work, so for the patients, we did not see, there was just one girl, 3 boys, 1 girl. She already had a very altered menstrual cycle because of all the drugs that she takes. So we were not able to assess if it changed her cycle because she was pretty much not having any period at all because the cocktail that she took was too great. But we did do a full hormonal panel, and we did follow them up with not only the research but the clinical side, and there was no difference. So we did not see change in estrus, we did not see change like in estrogen in the girls. We did see a correlation of testosterone and aggressive behavior in the boys, not in the girl. So we didn't see any side effect on this side. The other limitation that we have, as I mentioned, those patients are very, very severe. Most of them were nonverbal, so we could not do most of the traditional neuropsychological tests to assess memory and those things. So a couple of things that we measure that we questioned the parents was, we were concerned with olfaction because there's a big connection between amygdala and the olfactory bulb. And the questions we would ask the moms was, when you're cooking something that they like, do they still come to the kitchen? Because they don't talk, right? So normally what will happen is that mom is cooking and they would just come to the kitchen because they smell it. So yes, that was still happening, so we know that they were still having olfaction. But there was a lot of things that we were not able to measure with them. For the rats, there were no difference for the rats, no, the mice. There was no difference in estrous cycles. I measured throughout the whole thing with the females, no difference at all. I put that in the models. It did not have an effect. So that's for the amygdala part of your question. So for the DBS side effect part, yes, there can be side effects. There are irritability. When we're looking at those psychiatric targets, there could be irritability, agitation, there can be lack of sleep. But again, it depends on the target and the stimulation settings. So normally with the patients, what happens is that they come to see the neurologist very frequently at the beginning, and the neurologist adjusts the parameters. So they go home. In Canada, that's how it's done. The neurologist puts 2 or 3 different parameters on their system, and they can choose between those. So when they are home and they are feeling agitated or nervous, they can change themselves to the second parameter that they have. They don't have all the freedom. They have a few things to choose from. And then when they come back, they get that adjusted. Targets that are thought for movement disorder can have other types of side effects. So can have freeze of gait. There are a couple of other things that can happen. So yes, they can happen. We can modulate that. We can change that. It depends on the brain area. Thank you.
Other questions? Yeah.
Well, first, congratulations.
Thank you.
It was very interesting and quite novel, honestly. So I wanted to ask particularly about self-injury and the nucleus accumbens relationship, because I found it very interesting, particularly on how the codification of reward affects self-injury, particularly if the rat maybe codes for, I don't know, it sees that it fails, so maybe dopamine goes to the lower, maybe it believes, like in gamblers' brain scenarios, or like when the rats in drug addiction cases, when they get what they want, and when they don't get what they want, actually. So they might have a case, for instance, where the rat coding the reward in the nucleus accumbens affects how the rat self-injures in response, maybe to have an aberrant way of reward evaluation in the brain. I believe that's the question. Yeah.
Yeah, that's very interesting. That's a very good question. So there are 2 things with that, right? So we have the rodent world and the human world, so 2 different complexities. And it's not dopamine. We saw also that there is mu opioids that are being involved, so you have activation of the descending analgesic pathway. So there's much more to what I brought today, but the idea is that if the self-injurious behavior is being caused by an external effect, because they want attention or because they want something, like a temper tantrum or something like that, those types of self-injurious behavior you can treat with behavioral therapy. Normally, self-injurious behavior that are externally driven, so something else is happening and it's causing them to have that behavior, you can treat with therapy or medication. Those don't come to DBS because we can treat them differently. The ones that come for DBS are the ones that this doesn't work anymore. And then the theory is that that's the second type of self-injury, where the repetitive behavior is self-soothing, so reduces anxiety. The pain that they are feeling with the stimulation is leading to release of endogenous opioids, and that reinforces the behavior. There is the idea that they don't process inputs the same way, so the pain signal reaches the brain in a different way than other patients. So there's quite a lot of things involved with that. For the mouse model, there are 2 mice of models of ASD that have the behavior, the one that I showed and another one called Shank3. With them, we also think that this is what's happening because they are living in the same condition that all the other animals, and they have access to everything, so they're not deprived or anything. So we're not inducing, it's naturally occurring. And that's why we believe that the nucleus accumbens is having a important role in modulating the behavior, which does not mean that that's the only area of the brain and that dopamine is the only mechanism.
Yeah.
Okay.
Well, thanks. I want to ask again
Sure.
because I brought up another question while hearing you. And has it been already correlated nucleus accumbens activation, maybe even in more certain parts of hyperactivation with self-injury in humans? Can you correlate that? And maybe if there's like genes that alter expression in the nucleus accumbens, where maybe some people are genetically more prone to self-injury than others because they have those mechanisms being altered in the nucleus accumbens. Maybe we have a population that is more prominent for self-injury. So yeah, I believe it's very good what you're doing because I believe it can tackle those cases, which are very individualized, but also very particular